this screencast, our goal is to review the two types of electrical signals used by neurons and where each of these typically occur in a neuron. Neurons produce electrical signals which can transmit information. These electrical signals are generated by movement of ions across the membrane through ion channels, which changes the membrane potential. Note that these events do not appreciably change the concentration of ions inside or outside the cell. There are two types of electrical signals: graded potentials, whose purpose is to carry information from the input region to the spike initiating zone, also known as the trigger zone, and action potentials, whose purpose is to carry information from the spike initiating zone to the output region. Action potentials are sometimes also called spikes. Why is that? You're all probably familiar with how a single action potential looks when it's spread out. What can happen is when you have multiple action potentials occurring and the time frame gets compressed, is that each one of these individual action potentials just looks like a single line, and consequently they were given a nickname of a spike. So let's start by reviewing graded potentials. As we consider graded potentials, it's useful to think about a few questions. What is the function of a graded potential? How far does it need to be able to travel to accomplish this function? Where does it originate? As a result, at what location or locations in a neuron would you expect to find graded potentials? Graded potentials are only meant to be local signals. They're effective for carrying information over short distances, say about 1 to 2 millimeters, from the input region to the integrative region. This is because graded potentials are due to passive current flow in the neuron. This means that they decay or decrease in amplitude with distance from their site of origin. This is similar to ripples spreading from a rock or a leaf dropped in a pond. The ripples are largest closest to the site of initiation, and they decrease with distance. Similarly, graded potentials are largest where they start and get smaller as they travel away from that location. This is shown here for a graded potential in a muscle fiber. At the synapse, the graded potential is largest. So here's our synapse here, and here's the graded potential recorded right at the synapse. As the graded potential travels down the muscle fiber away from the synapse, we see that it becomes progressively smaller and smaller and smaller. Note that in this image, I've indicated that this graded potential is called an end-plate potential. That's because graded potentials are given different names depending on which type of neuron they occur in. For example, if you have a graded potential in the input region of a sensory neuron, it's called a receptor potential, and these are almost always excitatory. If we have a graded potential in the input region of an interneuron or a motor neuron, it's called a synaptic potential or a postsynaptic potential because it's produced through the activation of a synapse. These can be excitatory, an excitatory postsynaptic potential or EPSP, or inhibitory, an inhibitory postsynaptic potential or IPSP, depending on the type of receptor that's activated in the postsynaptic cell. For example, in the visual system, release of glutamate by retinal receptors can lead to an excitatory response in some retinal neurons via activation of an ionotropic glutamate receptor, and an inhibitory response in other types of retinal neurons via activation of a metabotropic glutamate receptor. The response is determined by the type of receptor. Finally, if we have a graded potential in the input region of muscle, it's a synaptic potential because it occurs through the activation of synapse, but it's given the specific name of an end plate potential, since a synapse from a motor neuron onto a muscle is called the end plate. Keep in mind that receptor potentials, synaptic potentials or postsynaptic potentials, and end plate potentials are all graded potentials. There's really no difference between them except that they occur in different locations in the nervous system, and therefore neuroscientists give them different names to help differentiate them. Next, let's briefly review summation or integration. Recall that a typical neuron receives about 1,000 to 10,000 inputs on average. The resultant graded potentials, both excitatory and inhibitory, travel to the integrative region, and can summate. If the sum of all the graded potentials, both excitatory and inhibitory, is above the threshold for initiating an action potential, one or more action potentials will be produced. So let's briefly review action potentials. As we consider action potentials, it's again useful to think about a few questions. What's the function of an action potential? How far does it need to be able to travel to accomplish this function? Where does it originate? As a result, at what location or locations in a neuron would you expect to see action potentials? The goal of action potentials is to convey information long distances from the integrative region all the way down to the synaptic terminal. Action potentials are produced or evoked if the membrane potential at the trigger zone reaches threshold. And technically, threshold is defined as the membrane potential at which there is a 50% chance of generating action potential. So if the sum of all the graded potentials just reaches threshold, you have a 50% chance of generating action potential. If the sum of all the graded potentials is above threshold, you have a 100% chance of generating an action potential. If the sum of all the graded potentials is well below threshold, then you won't generate an action potential. Note that action potentials, unlike graded potentials, do not summate. It's also useful to keep in mind that the images of neurons we see in textbooks are not often to scale. The axon of a typical neuron is usually much, much longer than shown, but it would be difficult to accurately depict this in most drawings. So here is a more accurate representation of a neuron, and you notice that the axon is much, much bigger than is typically depicted. And in fact, in this diagram, they're saying that 99.99% of the axon is actually not shown. So this helps to illustrate that the purpose of action potentials is to convey information these very, very long distances to the synaptic terminals for the next step, synaptic transmission. So once an action potential reaches a synaptic terminal, it can trigger the release or exocytosis of neurotransmitter. Binding of the neurotransmitter to receptors in the postsynaptic cell causes a postsynaptic response, for example, opening of ion channels. This allows ions to flow across the membrane and leads to a change in the membrane potential, which is called a graded potential, and more specifically, a synaptic potential. And these can either be, again, excitatory, an EPSP, or an excitatory postsynaptic potential, or they can be inhibitory, an inhibitory postsynaptic potential. That concludes the screencast on the types and locations of electrical signals. If you have any questions, please bring them to recitation or office hours.